It's nice to see everybody today, and I'll just give my relevant disclosures that what I'll be talking about today was funded by a Camper Pilot Award related to the disorder that we'll be talking about. I have some foundation funding from the two parent advocacy groups called the Yellow Brick Road Project and To Cure Rose Foundation to continue the natural history study. And we have a recent collaboration with Ovid Therapeutics, though there's no finances at this point, but we are starting to develop a relationship with this team. So when I see a patient in my office, I often will get to see them, and my role is really to think about, you know, what's the developmental concern, and that's really why most patients come to see me. And my job is really, I find it helpful to say, well, what is the diagnosis? What do we need to do to identify that diagnosis and treat that disorder? And a lot from a pediatric standpoint is related to anticipatory guidance. So what can I expect for the future for my child? And what ends up happening is these parents go home with a list of all these different symptoms or behaviors: developmental delay, anxiety, aggression, seizures or epilepsy, low tone or spasticity. And they end up going home also with lots of arrows that say, well, we're going to do therapy for this. We'll do prescription for that. We might be able to help this. Maybe medication can help with that. But unfortunately, we don't really know what to expect for the future because we don't have one all-encompassing kind of diagnosis for all of this. And that's kind of why I've been interested in the genetic disorder etiology, because it really can give us an idea of, well, we have these symptoms, but we do have a reason why they all can be all encompassed. And I think we're moving into the world of precision medicine saying, well, now that we know the genetic problem, why can't we just go in there and fix it? And obviously it's much more difficult than that, but it is really probably helpful, at least at this point. It really, I think, provides families with an understanding of it's not just a black box that we're working with here, and really we can better understand what the natural history of a disorder is. And so just to kind of go back to basics, a neurodevelopmental disorder, these are actually very common. 1 in 10 individuals have a neurodevelopmental disorder, and they can be any condition in the onset of an early developmental period. This could be a developmental delay, or it could be atypical development. And really it can manifest as many different deficits, meaning motor problems, communication or speech problems, problems with personal skills, adaptive skills, social relationships such as autism, academic problems such as learning disorders, and really everyday occupational functioning. And what ends up happening is, as you saw on the prior slide, patients end up having overlapping features, common comorbidities, and really the identification of a genetic etiology or a risk factor might help us identify a little more specifically what is going on. And importantly and interestingly, up to 30% of our neurodevelopmental disorders do now have an identifiable genetic etiology. And so in the US, a lot of these genetic disorders are given the term a rare disease. And as we know in this room, according to the NIH, there are approximately 7,000 rare diseases affecting up to 30 million Americans. And this is really 1 in 10 Americans. And this isn't just in America, those are NIH stats, but it really is a worldwide phenomenon. And unfortunately, because they're poorly or incompletely understood, we have fewer understandings of how to treat them, how to diagnose them, how to actually move forward in terms of treatment. And so most treatments we know require larger numbers of participants that are multiply replicated. And in the area of rare disease, this doesn't happen because you might have 20 or even less than that patients located in various locations around the world. The goal of my work has really been around natural history studies and following individuals with a disorder and monitoring them over time, collecting that health information to understand what happens naturally in this disorder. And this became important in spinal muscular atrophy, where they use natural history and natural history outcomes to really compare the natural history to potential outcomes and treatments that later became FDA approved. And now we have 3 FDA approved medications for a very common rare disease. And it's really important to have these natural history studies to facilitate that clinical development. And I think on top of that, as a clinician provider, to really provide families with an understanding of community and better understanding their disorder as a whole. And as you mentioned, today is Rare Disease Day. So there's a lot going on in the world about bringing awareness and appreciation and understanding of Rare Disease Day. And so what ends up happening is you have a rare disease that's initially described, and you have small nonprofit organizations that start out. And what ends up being the case is you have all these little tiny nonprofit organizations, and they're all doing the same thing. They're recreating the wheel in terms of getting their group together, building a natural history study, talking to sponsors, talking to researchers. And Simon Searchlight has really tried to allow families to collect information with any genetic disorder associated with neurodevelopmental delays or disorders, and really continue to collect that information in a safe, free manner. And then there's organizations such as Combined Brain, which is a consortium led by parent advocacy groups and really working with the clinicians and the sponsored programs. And both of these are nonprofit organizations that are trying to move forward in an organized way all together with each of these individual rare diseases. And so natural history studies are really critical for that process of drug development, and they have to really act in parallel to the scientific discoveries along with understanding what's happening in the gene. And so you really need to assure that you have collaborative attempts with the patients along with the researchers and the clinicians to make sure that we're moving ahead and treating things that need to be treated, and understanding that we're all on the same page. And so this is a schematic from this complex path from the Combined Brain Association, where they show the original gene diagnosis up until drug approval and all the steps involved. And so what I'm going to talk about today is most of my work in the last 5 years being at Columbia as faculty, the natural history study of this disorder, and also the results of our new proposal that was funded by the Pilot Award, working with my colleagues on a stem cell model to better understand and model the disorder, and hopefully move forward to clinical trials and drug approvals. And in 2006, we first published, myself and Dr. Chung, a report describing 6 females with predicted pathogenic variants, missense variants in the HNRNPH2 gene. This is a gene that's located on the X chromosome. And the clinical picture showed a girl with developmental delay. We had young ones such as the little girl here, and then that was our oldest individual in the group in her 30s. And some of these individuals had autism spectrum disorder, but all had developmental delay and/or intellectual disability. Many had muscle weakness, motor problems, some had seizures, and some actually had distinct facial features. We identified 2 hotspots, noted here with the arrows, where specific typos or missense variants within that genetic code was seen within the 6 girls, and they had the same clinical picture. And importantly, these 2 specific spots in the genetic code were predicted very important parts of the gene because they are highly conserved across various species. And interestingly, they actually were also conserved in other related genes called HNRNPH2, as well as HNRNPF. And so we were really interested about what was going on with those specific locations in the gene. And if you might be able to see, we'll go a little bit into this, but both of these arrows are located in a glycine YR rich region, specifically in this orange shaded area called the nuclear localization sequence, which is predicted to be important for shuttling that protein inside and outside of the nucleus and around the cell. And so we met with a group of eager parents. We had 5 parents from around the world who showed up on Martin Luther King Day in 2017 on an icy day outside of Rustberry. And we met with them, and they were interested in finding other people. And so they started a foundation called the Yellow Brick Road Project. And they started really sharing information with the world about their daughters and their personal families to try to find more patients and more families around the world. And since that time, we've had further in-person family meetings, as well as 2 virtual meetings in the last 2 years. Another nonprofit has also begun called Sakura Rose Foundation, and we're starting to really gain momentum with other collaborators in the field. And it was just 5 years later, about a year ago, we presented 33 individuals with the disorder, showing the clinical spectrum that involved other genotypes outside of that nuclear localization sequence, a wider clinical phenotype, and also included boys, which we originally presumed was caused fetal demise in males. And as you can see, some of the facial features we initially noted continued to be seen in individuals, even the boys. Acquired microcephaly, long, beautiful, thin lips, and almost almond-shaped eyes. Interestingly, many of these individuals had musculoskeletal findings such as scoliosis, hip dysplasia, and similar hand and feet findings like long, thin fingers and curled toes in older individuals. The MRIs that we gathered from many of these individuals were typically considered normal. However, there were some subjective findings such as underdevelopment of the cerebellar vermis, such as aplasia or hypoplasia, distorted cerebellar vermis, and decreased white matter maturation or a thinning of the corpus callosum, but altogether pretty unremarkable. And so now this is our most updated cohort, where we now have over 80 people that are enrolled in our natural history study. And we continue to hear from about 1 to 2 patients worldwide every month. And each one of these dots, or either a circle or a square or a triangle, represents a person. And what you can see is that that original hotspot in the 200 area, we now opened up here, and you can see that most of the individuals continue to have pathogenic variants in that specific sequence called the nuclear localization sequence. And there is an abundance at that one specific amino acid. But we are starting to note other phenotypes and genotypes that are kind of lying outside of that area, including frame shifts and other loss of function variants, in addition to those missense variants we first identified. We are seeing this in boys, and we also have reported it in a few inherited cases as well. We were really concerned 2 years ago, actually 3 years ago, when we shifted our meeting from in-person to a virtual meeting, and we were worried we were going to lose numbers and not be able to engage families and not continue to move forward with our participation. And so 2017 and 18, what I have here is the number of assessments and families and countries represented. We actually, in 17 and 18, met in person, but in 19 and 20, in 20 and 21, we actually shifted to virtual, and we have a 21 that's even more updated from here. We actually doubled the number of assessments and participating families in the setting of COVID, shifting to a virtual venue. And so we're recommending, or at least considering to the community, that we should have a paradigm shift, perhaps involving or engaging more families of rare disease using more of a virtual paradigm. And so my interests, I think I have to get out of this one. How does this work? Let's see. Uh-oh. Let's see if it'll let me do this. What I wanted to do was show you the motor disorder. Apologies. It almost let me do what I wanted it to do. The motor disorder is really what I've been interested in doing. Let's see. I'm not sure if it's letting me do this. Hang tight for 1 second, because I want to make sure I show this video. Can you see my slide with the video? I see a shaking. So I'll go into this mode for now. This is a little girl, and what we noted in almost all the individuals was that individuals of all ages have motor concerns. And so developmental delay with regards to motor skills was delayed in walking, weakness. A fair number of patients were actually diagnosed as cerebral palsy when they were younger. Many have low tones such as hypotonia, or high tones such as spasticity. And most of the group did report problems with coordination, motor planning, imbalance, and really gait problems. So you can see this little girl, it's very wide based and almost a choppy type of movement. Motor problems are a big issue in the group. And we certainly noted that from the very beginning. So that's what I was interested in. Tried, but it didn't let me keep me there. So I work closely with physical therapists and nurse practitioners in our group to really better understand and characterize the motor skills in this group. And what we're trying to do is understand different trajectories or subgroups to better understand the trajectory within the group as well as prognosis for this group. And so on the top is a measure called the Gross Motor Functional Movement, ADA, the GMFM, which is a clinician observed skill set. On the bottom, we ask for parent report. It's a parent report measure called the PDCAT. And in both of these, you could see there were two groups that we could distinctly statistically differentiate from each other. For example, those individuals who were nonverbal had lower motor skills than those who were verbal. And interestingly, while there is a pretty big statistical range here, and it was not statistically significant using the smaller cohort, there did seem to be a marked difference in the motor skills for those who had the genetic variants within that nuclear localization sequence and outside that localization sequence. So we're starting to better understand who are the more severely affected individuals in the group. And now we have about 3 to 4 years worth of data. And so we're starting to also ask ourselves what's happening over time. And so this is a modification of that clinician observed report, the GMFM, where it's 23 specific motor skills that we're asking individuals to do. We did this actually using telehealth. And so families would videotape their child doing the specific movements. They would rate it and we would rate it as well. And what we did was we wanted to see how an individual did over time. And so each one of these lines represents the average of a child over time. So the participants in yellow, for example, we had data for 2 years of those individuals. Those were kids who were 2 to 5 years old. The 6 to 10-year-old children were a little bit older. There was a group there. We had 3 years worth of data on those children. The purple is teens, so 11 to 20-year-olds, and then we have only a few individuals who are adult, who are greater than 21 years old. And in general, what you can see first is that as people get older, their skill set does improve and does increase. And importantly, they don't seem to be losing skills over time. We're starting to show longitudinally that they gain skills, albeit slow, but they are gaining skills over time. Usually they could acquire 1 to 2 more skills than they could the year prior. And I also wanted to highlight a really neat collaboration where we were looking more at gait analysis. So how do individuals walk? We used 2 different strategies. The first we published using an insole-based gait analysis where we had individuals at these family meetings walking down a pathway with these little insoles in their shoes. And we actually published to show that there was a big difference between those with the genetic disorders and typical developing peers, not unsurprisingly. And then this past 2 summers, we shifted to a virtual paradigm where one of our colleagues, Dr. Ardalan, took videos, home videos, and actually used machine-based learning to approximate the gait and the asymmetric gait to further characterize the clinical phenotype. And we're continuing to monitor their longitudinal acquisition of these skills. And so most of my work was really looking at the human, but what we wanted to better understand, how can we model this in a dish and really understand about therapeutics. And so we were eager to pair our work with that of our scientist colleagues here at Columbia. And so one of the powerful ways that technology now today utilizes is human stem cell programming. And this can be very easily used to recapitulate a particular disorder in the dish. And so this graphic depicts the idea. And what it's showing in essence is that you can take cells, whether that's a blood cells or a stem cell biopsy, I'm sorry, a skin biopsy, you can create cells from those individuals, and then you can try to manipulate those neural networks using different therapeutic techniques. And the reason why I was interested in this particularly was that the Simons Searchlight organization has been collecting and making iPSCs, or induced pluripotent stem cells, from this group. And so this was a low-hanging fruit that I was really quickly able to work with these wonderful colleagues on the bottom left. We've had a really exciting collaboration with neuroscientist Dr. Chris Recupero and our two stem cell experts, Barbara Corneo and Ajay Patel, to really build out our HNRNPH2 toolbox. And in order to build that toolbox, we've been using several patient-specific, patient-derived induced pluripotent stem cells from the Simons Searchlight. And what I have up on the right up here are, these are the 3 areas that we're targeting at this point with stem cells. The Simons Foundation generates these lines and is continuing to generate more lines in some of those more common variants, and we're continuing to work closely with them to obtain more samples. And very excitingly, these stem cells have been brought over to our lab, Dr. Recupero's lab. He's been able to grow the colonies and start neuroinduction protocols, making embryoid bodies and neural progenitor cells. And the bottom right, you can see that these are staining for neural progenitor markers. And we've just started to show the ability to start staining and identifying that HNRNPH2 protein in these cells as well. And so going back to where we started, we're really eager to continue to build out this toolbox using the induced pluripotent stem cells in order to really model this disorder in addition, and work closely in parallel to the natural history study. And we're excited to combine forces with the CTSA in starting to do some of these reprogramming and new therapeutic interventions. We recently combined forces with Ovid Therapeutics, in addition to continuing the close collaboration with the family advocacy groups, the Yellow Brick Road Foundation and To Cure Rose, to start looking at therapeutic interventions using these patient stem cells. And so just to finish up, the natural history study really is critical, and we find it's very important to ask families to continue collaborating with us and tell us everything that's going on. This disorder is associated with a spectrum of individuals. Some are more affected than others, and we really want to understand how to include everybody in that spectrum. And with precision medicine really continuing to emerge in the field of neurodevelopmental disorders, we're excited to keep collaborating with families and clinicians and researchers. And so this is a list of all of the wonderful people that have helped me get to here today. I want to say especially thank you to the CTSA for the pilot grant to really get us started on this. And we're really eager to move into the next step of potential interventions for our iPSC model. Thank you very much.